Hel- Good evening, everyone.
Good evening, Lynn.
Welcome to all of our audience on Saba and our live streaming audience around the world. Thank you for coming to the final night of See and Learn 24, the program that makes science fun. Woo! If you're at the live stream audience, you have to just bear with us a minute. We're having a wonderful evening with our people getting their last doses of the fantastic appetizer provided by Shea Booba Bistro. So first, a shout out to Claire Duco and the team at Shea Booba for hosting tonight and providing that great appetizer. Again, thanks to the Nature Environmental Policy Plan. So let's get started. We got a full night here. So before we get to our main program, we're going to do a little trivia to test the people who have been to presentations, maybe to allow everyone else to know what else went on this week, and to learn something. So our first speaker on October 1, it seems like a long time ago, was Erwin Winter, and he did a talk on endangered species. So the question is, how are conch able to move up to 22 meters per day? Woo! Who got it first, Emily? Rosa. All right. She was at that opening night. Yeah, they hop using a special foot. And the prize that you're getting is our sustainable cutlery made out of bamboo. Let's get away from plastic. Okay, our second speaker of the night was Eric Schneider, who came and worked with our fishermen and took us out diving. Ryan Hassel allowed us to do a dive on his fat at 2,000 feet. We only went to like 60 feet, but it was very cool. And he also, Eric, interacted with our fishermen, which was very cool for all of us learning how FADs can help scientists as well as benefit fishermen. So the question is, which of these is considered a FAD, a fish aggregating or attracting device? A floating log, B, sargassum seaweed, C, a chain with a buoy attached, or D, an artificial reef, or E, all of the above. Oh boy, how are we going to do this, Emily? Okay, Michael, our board member, says the first person to get the answer was? Oh, that it was Mike. Oh, it was. All right, Michael, we're going to defer to Koska. All right. Koska's right here. So let us finish reading the question for the audience, and then we'll try to figure out who's the first one. All right, we're going to make it more difficult. Our next, all of the above. Okay, our next was Jan and Aglaya, insect specialists. And, a few people went, well, insects, is that going to be interesting? It was awesome. Their presentation was great. Their field projects were even better. Their work with our school children was amazing. And the question is, what new order of insects previously unrecorded on Saba did Jan and Aglaya find? A, web spinners, B, beetles, C, earwigs, D, dragonflies. I see the first hand. I don't think so. Oh, we've got Mia in the back because she learned at school. C, earwigs. That was the same answer. Okay, Nolan. Emily, you wrote this question. Okay, sorry, Nolan. We'll get the next one. The answer is that goofy-looking web spinner. Okay. There you go, Nolan. Thank you. 2 winners. Round of applause. Ellison Robertson, misspelled there, whoops. We got to dive the Saba Bank because she wanted to go out there and take algae samples to see how they compared to other areas, and she's a toxin specialist. So very cool field projects, fantastic presentation. The question is, where does the ciguatera toxin come from? I think I gave that away a little bit. Okay, we got the first winner, the dinoflagellates in algae. We could have done an Alex Trebek on that. Okay, bonus question. What environmental factors affect the toxin load? She said global warming. We can see a hand. Anyone else? Erosion. Elevated nutrients. Anyone else? Cruise ships? I don't think so. The answer is light waves, temperature, species of algae. I told you you're going to learn. Okay, Aaron Popke gave us a fantastic eye-opening presentation on coral microbes and did some sampling on the reef, and also, of course, worked with our school children, and you can see those great photos of what they were learning. So the question is a long one. Stony coral tissue loss disease, also known as Skittle D, is the most aggressive coral disease in the Caribbean, and until recently, antibiotic applications were the only known treatment. What new treatment not only improves diseased coral health, but can also prevent disease? Okay, so A, injecting bleach, B, yogurt, C, probiotics, or D, ivermectin, and all of the above. Okay, I don't know how fair this is because Camille is our coral person at the Conservation Foundation, but she was the first one. Yes, and the answer is probiotics. So there you go. Okay, which of the following best describes the concept of reverse genetics? A, studying an organism's traits to identify the underlying genes that cause them. B, inactivating or altering a known gene to study the effects on the organism's traits. C, using DNA sequences to create an organism from scratch. Or D, crossbreeding organisms to study how genes are inherited over generations. I heard B, I heard D. I think Sherry was the first one to say B. And I heard a D, but I never heard an A. But the answer is A. Oh, A!
No, wait, go back to the previous slide. That was
You are trying to cheat.
Okay, it's a long month. We do make mistakes. All right, there you go. Good catch.
Was that one of the quiz questions?
Yes, it's final exam. Okay, here we go. Mark Butler, crustacean restoration. And who learned how to do the lobsterina? If you were on the boat, you had to be on the boat for that. Okay, so what are the three main types of herbivores, grazers on Caribbean reefs? You got to name all three. Come on, let's get a good answer.
Urchins, whelks, and crabs.
I think you're right. Oops. Urchins, whelks, herbivorous fish. You could probably say you need to add a fourth there, but yeah. Okay, Gina Hendrick came here. She came back. She was the master's student with Paul Sickle last year, working on her PhD, and when she came back this year, She came back as Dr. Gina, and she specifically studies fish parasites and had to do a whole lot of dives to figure out her project and pick things up here. So the question is, which fish are most affected by nathid isopods? I heard parrotfish. I heard grouper. I heard grunts. You got to help me out, Emily. I can't watch the crowd. What did Sue say? Damselfish. Were you the first? Okay. It's grunts and snappers. Okay. This is Bob. Gets it with grunts. Bonus question. What do these species, Bob, what do these species have in common? Grunts and snappers. Ah. Oh, Femke got it first. Sorry. They migrate from the reef to seagrass beds at night and back to the reef again in the morning. So, yeah, well done. People are listening. That's good. Okay, we fondly referred to these two as R&R. Roseanne and Roel came from the Netherlands to talk to us about springtails. And we were like, okay, what are springtails? But again, all of our scientists are so passionate, we all fell in love with springtails and did incredible field project, a nice sketching exercise activity. So what can you find, where can you find springtails? And Jesse got it? Shelley, all right. Prize to Shelley. All of the above. Yeah, they're everywhere, including in Antarctica. Okay, we're almost to the end. Alex Sloan came from Puerto Rico as a seed dispersal expert. He met with our folks doing reforestation on the island. Lots of people did a really cool field project, as you can see the photographs down near the tide pools. And he did plant pressing with kids. So the question is, name 3 ways in which seeds are dispersed. I want to see a hand. Who wants to name all 3? Nolan, tell us. I couldn't hear him totally. Dig a hole, put a seed in it. Okay, that one's definitely correct. Animals disperse them by eating them and pooping them. And the third? Wind. Very good. How about a fourth? Woo! Now, does that show we have impact with school children or what? Learning outside the classroom is very impactful. Thank you, Nolan. Okay, Lars was just a few nights ago, the tall guy from Curacao, also talking to us and teaching us different things about coral restoration. I don't want to give anything away, so here we go. Which 2 kinds of coral seem to be good neighbors to each other, at least in Curacao? Come on, Camille. Somebody else. How about somebody who's not working in corals for a living? Come on, you went to the presentation. It was about neighborhoods. Which 2 corals together work really well? Okay, Larry.
Fire coral and sagworm?
No, but you're kind of giving it away. Somebody else? Michael? I hear staghorn again. Not brain coral. Not lettuce, not elkhorn. There's a dive site here named after it, that type of coral. Also known as Lou's ladder. Sherry, yes. Porites coral, and also known as finger coral, and fire coral. So there you go. Okay, just the other night, Kimberly Stewart, who's in our audience, give us a wave, Kimberly. She did one of the most engaging kids field activity out of the classroom. It was wonderful. She gave a fantastic presentation, but let's see if you were paying attention at the presentation. What physical feature do sea turtles have that allow you to identify individuals from photos? Oops. Shell pattern. They're yelling in the back. Distinct scaling patterns is the correct on each side of their face. So this is a non-invasive way that we're going to encourage snorkelers and divers to help us really track where we're seeing turtles throughout the Caribbean and the world. Okay, so here we are. We don't have a trivia question yet because he's tonight's speaker. So a round of applause for everybody. I think that was pretty good of everyone remembering what happened this month. Do I have another slide, Emily? Start here? Okay. We are closing out See and Learn 2024 with another pun, with another jawsomely fantastic presentation on sharks. If you don't know, my colleague Emily is the punster of the organization. Some are cornier than others. Erwin Winter is a scientific researcher at Wageningen Marine Research and the Aquaculture and Fisheries Group of Wageningen University in the Netherlands. He has 30 years of experience in studying fish behavior in a wide range of water systems. He uses a wide range of techniques to measure and understand behavior of free-ranging fish in the wild. For example, mark recapture experiments, small acoustic transmitters in combination with extensive detection station networks, and archival tags. With individual tracking of fish, he studies the impact of anthropogenic, also known as human, pressures on behavior, what consequences this has on population levels, and what actions can be taken to safeguard or restore fish populations that are in decline, such as sharks, Atlantic cod, European eels, salmon, and lampreys. Please welcome Dr. Erwin Winter.
Yep. So, good evening, everybody. It's really nice. Yeah, it's really nice to see so many people turning up. I'm really glad with that. I hope I can tell you an interesting story about all the shark studies we did in the past over 10 years in the Dutch Caribbean. Is it? Oh, it's yeah. Oh, sorry, how to turn this on? Oh, there, okay, sorry. Yep, now it's working. Get some sponsors. Yep. So I'd like to tell you about sharks in the shallows. I'm going to talk mainly about the studies we did on sharks that dwell in the shallow waters surrounding islands in the Dutch Caribbean, with a special focus on Saba and the Saba Bank. And yeah, well, I was already introduced, so I don't need any further on that. Reef-dwelling sharks, they are in peril worldwide. The numbers have depleted enormously all around the world. There are some areas where they're less depleted, but also in the Greater Caribbean, it's a pretty bleak picture, I think. If you would dive here 600 years ago, I think you would be surprised by the amount of sharks you saw in the water. It's no comparison to what we can see today. And there's differences between sites, but overall, that they're having a very a tough time. And that's mainly overfishing, a long history of overfishing, really depleted sharks. They are very vulnerable to overfishing due to their life history strategies where they grow old. It takes a long time for them to mature. They have only a few young offspring every 2 or 3 years, many species. So they cannot compensate any extra mortality in the populations. So they are very vulnerable. If the mortality rates increase already a bit above natural levels, then they're already in trouble. So also recreational fisheries and non-release. Luckily, there's more and more tendency that recreational fishermen are much more careful with their catch and do catch and release fishing. But also, let's shift around. So habitat loss due to coastal development made a lot of coral reefs lost, and of course, more recently, and a big threat in the near future is climate change and bleaching and changes in coral reef systems. I'm really concerned about developments there. And we heard it also in earlier presentations. So that's going to be a big challenge in the future, already ongoing. So today I would like to take you along on different studies we did in the Dutch Caribbean. So we have the Windward Side Islands here on which are St. Martin, Seba, and Statia, and here St. Eustatius, so we call it Statia. And the Sable Bank, and I'll show some mainly focus on that area that we enlarged here, and you have the ABC Islands, the Aruba, Curaçao, and Bonaire, where I will show also some work with the BRUF studies that have been carried out there, and the shallow waters here are mainly comprised of 3 different type of whatever coral reefs. You have seagrass reef, and you have more sandy habitats. Of course, there's a bit more differentiation, but these are the 3 main habitat types that you can find in the shallow waters surrounding these islands. One technique that we use to assess the status of sharks is BRUFs, baited remote underwater video surveys. You have a frame with 2 cameras or 1, but if you have 1 camera, then you only do presence absence. But if you have 2 cameras, you can also, because you have, you can also estimate the length of the sharks that are in the footage. And then there's a pole with bait, a bag with bait to attract sharks to enhance the chance that you would see sharks coming in from a wider area. So also more rare species can then be seen. And there was since 2012 until 2017, first round of BRUF surveys was carried out in all 6 islands and also on the Sable Bank. It was initiated by a former colleague of mine, Martin de Graaf. And I'll show you a clip of a juvenile tiger shark on the Sable Bank. It's from Ton Stoffers. He's in the audience also. It's from his work on the Sable Bank. So this is a juvenile tiger shark. And I would make a short sidestep that sharks use many senses to that shape their behavior that trigger all kind of behavioral traits. Real long distance hearing travels the furthest, but they can only hear low frequency sound. That's high pitch sounds they cannot hear at all. The threshold for hearing sounds is at a relatively low frequency sounds. Very important in foraging is smell. They can sense minute concentrations from long distance. So with the BRUF studies is what we use to attract species from different from a larger area. And these BRUF studies, they're always done in the same way all around the world. Typically 1 hour of deployment, so that we can always compare the results between different sites with each other. That we have that the base for that data is the same. So but it can be that some sharks are attracted from further away, some species, than other species that are more from locally. Yeah? So you should be careful with, if you have BRUF data, that you look at the abundance, the amount of individuals you see, that you cannot directly translate that to how many sharks. If you see more tiger sharks than Caribbean reef sharks, you will not say that tiger sharks are more abundant. Could be that they also come from a wider circle. So you have to know and understand also their behavior to interpret that. But what you can is compare the relative species, the relative abundance between different sites for a particular species. That you can compare well. And also species richness, it's a nice way of assessing the diversity of species that you can find on a site. And then, another thing, that if you look back, you see it biting in the camera, and that is related. We often see that they are very curious to the camera, and that is to the smallest site sense they have. They have the special pores with ampullae of Lorenzini, where they can sense minute electrical currents, and they use it to detect electromagnetic fields and electric currents that are, for instance, when you have muscle activity, it will send out some very small electric signals. So they can, in the dark, they can detect a predator up close approaching them. They can look underneath the sand. If there's hard activity of prey buried in the sand. For instance, hammerheads have really wide heads because then the difference between the pores is highest, so they're even capable of detecting even smaller electro currents. That's why they have such an odd-shaped head. So this special sense unique for elasmobranchs, that the sharks and rays. It's not always going according to plan, as this very feisty nurse shark shows, is ripping the bait back completely off. So this, the attraction of this site became a little bit less after this nurse shark visited the setup. And here's an overview of the results that Twan, during his master thesis study, comprised. So you can see the whole. There were drops of in the whole Saber Bank. It's a huge area. It's about I think 50 by 30 kilometers, something like that. It's a huge. It's one of the biggest sunken atolls in the world. You can see Saba there on top. It's only a tiny speck compared to the Saber Bank. And here we, the Caribbean, as in most sites, Caribbean reef shark and nurse shark are most prevalent species. You see that most often. And you see now tiger shark is also seen occasionally, and here we also saw then a blacktip shark. That's a more rare treat in these areas. They are more common in the Bahamas, for instance. And here an overview of all, so on the y-axis you can see the percentage of drops where they were seen, the fraction of drops. So you can see that there was also a lot of drops without shark sightings in this area. So you can see that in Bonaire and Curaçao we had lowest numbers of sharks compared to all the other islands. And in St. Martin, Saber Banks, in the Stations, and Aruba, we had Aruba was especially also quite rich in species. Nurse shark was by far most abundant on the Saber Bank, and Caribbean reef shark was similar abundance present in 5 out of 7 sites. That's the most commonly observed species. But we also saw some other species there. But there are only a few of them in the data set, like great hammerhead, silky shark, sharp-nosed shark species. We could not determine the exact species from the footage. Bull shark and lemon shark, but these are more rare occurring species in this area. And this complete data set was used to feature in a global meta-analysis, comparing all kind of sites worldwide. We're also with Van and Martin de Graaf cooperated as co-authors in those publications. And yeah, we could see that there was in the BRUF data we saw for the two most abundant species, the Caribbean reef shark and the nurse shark. That's on the right hand side. Most of them that we observed in the BRUF data were juvenile individuals. You can see here's the length, and the gray is in that. There's some variation when at what length they mature, but in these somewhere in this gray area they mature. And you see that, like for instance, nurse shark, they can take females 16 to 18 years to mature, and then they're 2 meters 20 before they are. So it takes a long time for them to become mature females. So adults are more sparse in the BRUF data set, but it also highlights that these areas are important nursery areas and for young sharks to grow into adulthood. And we also saw that even within these juveniles, there was also we call it ontogenetic shift, but during their development from a newly born neonate to juveniles and then adults. That is the ontogenetic trajectory. And we saw that the youngest juvenile, they occupied shallower habitats than the total length. That in the shallower was on average smaller than the average length in the somewhat deeper areas. So the young sharks during the juveniles, they also shift to a bit deeper reef waters during their development. Right now there's a new global FinPin 2 follow-up action from the earlier year worldwide BRUF study, and we're again joining in together with Twan and Tadjo Barefoots and more people, but they play an important role in this. And then and within this follow-up study, we do the Netherlands and France side of Sint Maarten, Seba, Statia, and Curaçao, and they selected mainly sites where there's a protected part and a non-protected part close together. So we can, on the whole, we can see what is the impact of protected areas compared to 10 years ago. And the unprotected areas, is there a lot of difference at sites, or is it so that will be analyzed in the course of next year? We're now gathering the data, and this is fresh footage from Sint Maarten from last week with a very beautiful. That comes. It's a newly born tiger shark with a beautiful striping pattern. When they're really young, when they are born, they are about 50 to 70 centimeters long. They can have quite a lot of young. The tiger shark is different than other large species in that it has more smaller young than, for instance, a great white that only has 2 youngs of 1.5 meters. But this one is very young. It's I wouldn't be surprised if it's only a few weeks old. And the striping they lose more or less when they age. So we see in adult tiger sharks, they're much more vaguely colored than these juveniles that are very brightly striped. They're magnificent animals. Another type of data we use in close cooperation, for instance, with Sea Seba, this observations from scuba diving. These data sets were built in cooperation with Lynn, who will host this See and Learn study. And with this citizen science data and the scuba dive set, we have a full record of all the dives, if there were sharks seen, and which species. And that really helps also, and we have then much more sites available because and we can also look more at seasonal trends, how things vary throughout the year. So that is a very nice additional data set that we could use for SEBA, and that's not on all islands even easily accessible. Those data, the degree of cooperation varies between islands. And what there was. This is the trend for data 2012 to 2020, and the probability of presence per dive. And we saw the green is the Caribbean reef shark, and red is the nurse shark. For nurse shark, we see a small increase, but for Caribbean reef shark, we see a steeper increase. That looks very promising, especially if you see how poorly in a poorly state these reef sharks are. But we have to be careful in interpreting this because other things it might also underlie or play part in this pattern. For instance, in this period also lionfish became more abundant. There was more lionfish culling, and that and with the zookeepers, you can safely do that. But sharks find that interesting, and perhaps they are also more willing to approach scuba divers. So that could also be part of the explanation why more sharks are on average seen per dive than in the start. But I hope that it's truly an increase in population level underlying these. That would be a real nice positive sign. And then I would like to take you along with telemetry studies that we did, acoustic telemetry, where with acoustic telemetry we build a network of listening stations. So these are the listening stations, and they're just deployed with some concrete blocks and a float on a rope and placed on the bottom by diving. These listening stations, they can operate 15 months on their own, on battery, on the battery, so they're a standalone system. And they're passive listeners, but they're filtering at 69 kilohertz if any signals come in. And we use small transmitters that we implant in the body cavity of sharks. And these small transmitters, they emit a unique signal for each tag, worldwide unique. So also if a shark shows up in a complete other area or other array, we will see that was tiger shark John tagged at St. Martin in 2015. And these tags, the programming we used, they last for about 5 years. So we can follow individuals then for 5 years. And the new generation of tags, we can even follow them for up to 10 years. So it's becoming the technology development goes quick in that as well. So underwater, there's no GPS available. The GPS signals, they die out in just a few centimeters of water. They're used as underwater. So always, when you track fish, you always have to use other techniques and more indirect techniques to get their behavior visible, to make their behavior visible for us. So we build a network around St. Martin, the Sable Banks, the Stasius and Statia. On average about 32, 36 detection stations in total. And here we started with a pilot in 2014, November 2014 at Seba. That was very successful, and then within the Save for Shark project we could expand the network. And you see some gaps, like for instance here in St. Martin in 2017, in September with Irma, we lost with the hurricane, category 5 hurricane, we lost all the receivers there. We only found 2 back with these big concrete cloth. One was moved 800 meters from where we deployed it, and it was full of water. And they're really powerful. They're really sturdy equipment. So you can imagine the forces that happened underwater there. So it took a little while before we were back in business on St. Martin with the network. And if you want to tag sharks, you have, of course, have to catch them. In the early days, we worked closely together with fishermen on Statia Still. The first sessions were Michel here on Seba. We caught them with a rod and line, and we bring them alongside the boat. And then if it's a smaller shark that we can handle, we take them on board, because then you can do a quicker procedure than when you do it in the water. But if it's a larger shark, we will fix it with ropes, flip it upside down, and then sort of magic happens. That's unique to sharks. That if you flip a shark upside down, it becomes in a sort of sleeping trance-like state, and we call it tonic immobility. It's still not clear how that physiologically works inside, but we do see that they don't respond to any pain stimuli. So if we have them fixed alongside the boat, and I make a small incision in the body cavity to insert a tag, you see no response on that. We then suture the wound, and then because I always like to work with long-term studies, I always like to work with internal tags, because it's a bit of a more invasive treatment at the start, but it heals very quickly, and then there's no harm anymore. But external tags, you always have an exit wound all the time, small exit wound. You have fouling that can grow on it. So it's if I have the choice for long-term studies, I prefer to work internally in the body cavity. And then, after then they're released, and they can gather data for us. Then we sit back and relax, and now and then do a readout of the detection stations. Just a funny detail here. You see here these fins, and you see two penis-like organs. Those are called claspers. And with adult males, they're much longer than these fins. So if they're much longer and they're calcified, they're really hard tissue. And with the juveniles, they're much smaller than the fins, than here. So with males, you can always see if it's an adult or a juvenile. If you're diving, also you can also distinguish males from juveniles and females. But for females, we cannot. They don't have these claspers, only these two fins. And there we cannot distinguish between juveniles and adults, and there we use a length key to determine whether it most likely is a female or an adult. And so within the telemetry study, we focus mainly on 4 species: on the Caribbean reef shark on the top, the nurse shark, the silky shark, and the tiger shark. I'll come back to the silky shark later. And here you also see, similar to what we saw in the Bruff data, that most of them are juveniles. So you can, these purple, those are the length that they have at birth, so when they are born. So you see that for instance, we have some neonates here, and also with tiger sharks, we had some very small individuals that were newly born, and then they had juveniles, and then in the blue band, that's the band where they grow, where they mature, get adult. And these were all adults, so they are still in the band, but we could see, for instance, for the males, you can identify them as adults. So for tiger sharks, we have a group of about 7 adult sharks in it, and for here is also part of them are adult. And nurse sharks were close to adult, but they are really big. They need to be really big before they mature. And so also here we have a lot of juveniles in the study, but we can follow them for 5 years. So during the course of the study, they can mature, and so we also have adult data in our data set. But then we have to take into account how fast they grow. So these four species, we had in total 100 sharks tagged, 52 Caribbean reef shark, and on the other three species, around 15 we are in the study so far. And here you see each line is an individual shark. So the top panel is the Caribbean reef shark, then the nurse shark, and then the silky shark and the tiger sharks. We saw that, and the last year's data is not in yet. And especially last year, we tagged quite some tiger sharks, so that they will come. They're not in the data yet, but they will come later on. And you can see that, and the color is, it's a bit small, but is what island? So Saba is, this color is Saba, around Saba. Green is the Saba bank. You have the Statia is light blue, and purple is St. Martin. And you can see that they quite a lot of them stay for a long time in the same area. You see some species that change, like this one that moves from Saba to the Saba to Statia and back. Here we also have some. Those two are very similar in pattern. It looked like they teamed up, but then later on, this one was seen there without its. That might be that they teamed up. They have very similar detection rate. It's a busy graph, but I just want to show you what kind of data we then have. This is an example of one adult Caribbean reef shark of almost 184 centimeters. It was tagged on the west side of Saba, and in the 5 year we could follow it. It was only detected on the west side of Saba, and moved around here, but never on the other side. So a very local individual, and it's a pattern that we often saw in Caribbean reef shark, both in juveniles and adults, and also in juvenile nurse sharks. They're also very. They have their own place where they stay for long times. You could add. We have had more detections during night than during the day. This is the hours of the day, and this is the number of detections. So we saw them more at night than during the day, and that you can also see that here. That this is the date, and this is the hour. So you can see here that the dusk and the dawn and dusk patterns in that, and this one moved between different stations. It was most of the time here, around this area, but it also moved sometimes to the pinnacles or Shark Shoal, so even within that already confined area, it was mostly present in around here. So we found that in many individuals, and it's still a bit speculative, but we see team them up. We see them also swim together. There doesn't seem to be strong territoriality, so we don't think that they occupy territories that they defend against others. But what we do think is that coral reefs are very complex habitats. And it really pays off to get very familiar and know your area really well to be an optimal forager in that area, and that naive newcomers have a disadvantage in that. That seems like a likely explanation underlying the data we found so far, but it's difficult to prove and test that. It's an idea I like that fits the data quite well. Just an overview. So we have for all sharks we have we saw them in St. Martin, the Saba Bank, and the Saba Bank is huge. We only had the drop-off zone on the east side covered. So what they do on the rest, we don't know. Silky sharks, they were especially located here. We only saw them here, and we also caught them only here in the area near Poison Bank. And one of these silkies showed up twice at the coast of Stasia, and it was the only station where you have deep water adjacent to. The other stations are more on the shallows, and this one was with a steeper drop off, and that was the only station where it popped up twice in different occasions, and being back on the Saber Bank in between. This perhaps I won't go into detail here, but what I would like to show is the site fidelity and residency. So here we have site fidelity. So how strongly do they stay on the same in the same area? And here is the residency. Is that and which % of the time during the first to last detection were they seen? So if you are very local and you stay there all the time, then you would end up here. So the Caribbean reef shark and the juvenile tiger sharks we had, because the adults are not yet in this data set, they are more in this corner, so they're very localized behavior, and nurse shark quite close to that. But we saw that for the silky shark, they were much less present, and we think that they also occupy the pelagic deeper waters next to the reef, so that they spend part of their time, and that of course it was not covered with our detection network. We also looked at differences between the zones. So we here we have just for Seba, where we compared the acoustic telemetry, the citizen science, and the this is the Bruff. This is Bruff. Yeah. And you could see that that between especially the recreational fish, where no fishing is permitted, that for Caribbean reef shark is the most frequently used zone around Seba. We saw some nice. For instance, this is one individual Caribbean reef shark that from Stasia. It was tagged on 20 May 2019. On 10 July, it was all the time around Stasia for 2 years in a row. And then on 10 July, it made an excursion to the Saber Bank. It stayed there for a week. In 24 hours, it swam there. And then in 15 hours, it went back to the exact same spot on Stasia. It was a very directed back and forth excursion from their very local home range to the Saber Bank. And we also saw that for some individuals on Seba, similar short excursions to the and also there, it's speculative what is underlying, but we saw it more in adults and in juveniles, and the juveniles we saw it in might have been already adult because it was later in their detection series. So we wouldn't be surprised if this is related to mating excursions, that they gather in certain areas for mating. We even have some even larger scalers, like in the US Virgin Islands, they use the same technique worldwide. We use the same technique in acoustic telemetry. So they have networks there, they have their own shark studies. And 3 of the sharks, two tiger sharks, one went to St. Martin and then to Stasia from the US Virgin Islands, and one went to the Saber Bank and Seba. But also a nurse shark moved there, and they were also quite surprised that the nurse sharks moved for such long, over 200 kilometers to the Saber Bank. But in California, in Florida, they also found that adult nurse sharks are much more mobile and can migrate over much longer distances than previously thought. And that's also what we see. We had also one nurse shark we thought we tagged on the north side of Saba. We released it. I only had one detection. Didn't see it for 3 years. So it was outside our area, but there's a lot of white gaps, of course, everywhere. And then after 3 years, it turned up at the Saba Bank. And then half a year later, it went to Saba. So also that animal moved around quite a bit. So it's still a bit anecdotal what we have for adult nurse sharks, but they do show similar patterns of what they found in Florida as well, that they can move over much wider areas. Now I'd like to go back to the silky sharks. That's Poison Bank. It's this protruding with deep waters all around. That by accident, while we were working there with snorkeling, there were 10 juvenile silky sharks surrounding us. It was lovely, a lovely snorkel. And we tagged here also silky sharks, and we mainly see them in this area, and one seen a few times in Stasia. And silky shark is only using reef habitats as juveniles. When they're adult, they go out in the open, wide open, pelagic, but they're highly endangered. Numbers of silky sharks, they really plummeted worldwide, so that's a species that we should really be careful with. And the Saba Bank, this seems to be a particular hotspot for juvenile silkies to grow up, and they're lovely. They're very elegant sharks. They have a more rounded dorsal head, and not so pointy as a Caribbean reef shark. So that we. For the silky sharks, we also used tags with a depth sensor, so we could also see at what depth they are, and we compared that. We also had a group of juvenile Caribbean reef sharks that we used tags with a depth sensor, so we can compare how subtle differences in habitat use between these two species that live together on the reef, that share a reef, but they do it in a different way. We also did some sample tissues we took to do stable isotope analysis, and then we can see whether they feed more on pelagic prey or more on benthic prey. So that will also help us in how the differences between these two species are in sharing a reef for growing up. And then for tiger sharks, this area is also has a special is also special for tiger sharks. This is a neonate. This was 78 centimeters, so we call it St. Martin. It's a beautiful. Similar size, I think, what was on the Bruff, but it was a different one because this was 2 years earlier. And the adult sharks, larger sharks, we of course don't get on board. That we keep on the side of the board. And we found mainly on the Saba Bank and St. Martin that they are important also for the youngest life stages, that they show up there. And last year in November we were also tagging sharks, and we had a session on Moonfish Bank, just a bit northeast from Poison Bank, where the silkies were. And here we had. We caught 4 adult tiger sharks. We had 1 adult tiger shark that got loose next to the boat. We had several hooks. There are really strong circle hooks. They were completely bent out. So there were even bigger tiger sharks. That you need a lot of force to make that happen. So that what was very ominous is all the sharks we had next to the boat, and also the one that got loose, they all had fresh bite marks on their head. You can see here with the fins, and and yeah, some were more bitten than others. But this is also a clear sign that we stumbled upon a mating aggregation of tiger sharks. So also the saber bank is used for mating by tiger sharks. But this is it's very difficult to determine that. So it's more a lucky observation that we had, but it was very impressive to see that. So yeah, I would like to run a few more concluding remarks with you. So we found very strong residency, especially also for Caribbean reef shark. We didn't expect them to be so to have such small home ranges and be so fidel to a small area. For juvenile nurse sharks, it was known already that occurred, but for Caribbean reef sharks, it was a bit more surprising, and for adult nurse, it was the other way around. They are more mobile than often thought up front. And juvenile tiger sharks, they roam in a certain area more widely, but they can stay. We had juveniles that stayed for more than a year on the south side of St. Martin, so they can also linger on a certain area for quite a long time. The saber bank is definitely a special area for hosting, because it's large. It can host larger populations, but also it features as a hotspot for juvenile silky sharks, and yeah, we stumbled upon a tiger shark mating aggregation. Well, I also showed you these back and forth excursions, but they're very rare. That takes sometimes an individual that spends years on one side and then makes a directed excursion and goes back. So you need long-term data sets with quite a lot of individuals to find such phenomena. You can easily determine their habitat use, but these more rare events, you need long-term studies and denser data sets to identify those. We saw clear connectivity between reefs that are separated by deep waters, more than 500-meter deep waters, even from the US Virgin Islands. And if you see how many islands are around us and reef systems, that we already had quite some exchange between where the few places where we have detection station. That must be only the tip of the iceberg. I'm sure that a lot of under the radar movements occurred that we didn't see because we have no networks there. And yeah. Because they have such small home ranges, even small marine parks, they can protect individuals. They can safeguard individuals for fishing, but of course, you have too few individuals to sustain a population. So you need then a patchwork of smaller reserve, but with good connectivity to also sustain the population, and preferably you would have more large-scale sanctuaries, like for instance the Yarari sanctuary that has been installed around the Saber Bank and Statia and Saba. So that's what I would like to share with you this evening, and I'm happy to take questions if you want. Yeah. Yeah. There's. I'll repeat the question. Yeah, the bite marks, are they from males only or also for females? Well, we saw them on both males and females. We had more males than females than that day. But yeah, usually it romance in tiger sharks is not very subtle. They grab each other, they force each other on the side, and we also think that tonic immobility plays a role in that. That when a female is turned upside down, forced upside down, that it gets more docile, and then it's more easy to mate with her. So it's yeah. There's not yet a Me Too for sharks, but I'm sure that if it comes in place, we get some registrations. But there, yeah, these fresh bite marks, and also in other occasions, it's been documented that it can get quite roughly. And they also always have a lot of healed scars that from earlier. But these were all fresh. They were freshly and they're still bleeding. Yeah. But, yeah. Hey, Dalia. Dalia also took part in the studies on the Saberbank when she was officer there. Yeah.
Thank you, Erwin. My question is based on the last, well, I guess it's not really confirmed, but we've always been talking that St. Martin can be the area where they go give birth to these baby, I'm talking about tiger sharks, and then the Saba Bank is where they're doing the mating. But how long does it take for you to have a confirmation of this, or what else do we need to do to make sure that we can get these confirmations and then do the right steps to protect these transboundary species?
Yeah, I think that also the Saberbank is used as that also their pupping takes place. I think both St. Martin and the Saberbank are used for pupping. I think for one thing I didn't show you because I was not involved in that as well. We were also tiger sharks equipped with satellite tags, because the tiger sharks, they with a dorsal, they surface sometimes, and then you can use GPS data on that. So there are some tiger shark tracks, and they can really roam for large areas. I think if you have a sufficient amount of, if you have a combination of showing that neonates occur in areas, you could with bruffs very well, and you have for the adults sufficient amount of adults swimming around, both males and juvenile, and males and females. I think then you can derive what might be important mating areas where they come together. But I think you need. Yeah, you need a certain amount of individuals to track it. Well, 100 is that's a you need huge budget then. but yeah, but I would say the data so far is we have very nice patterns. I was very impressed by the patterns. There was also some methodological problems, but I think if you have, let's say, 30 individuals, something like that, then you have a good chance, and we might. What we see in other species also is that it's often it's not a yearly phenomenon. They mate sometimes only once every 2 or 3 years, and then. And with the satellite tag, they don't last that long. So, if you have a good idea of where, you can also use a combination of tags, so that you do satellite tags in combination with a long-term acoustic tag, and places where you expect them to mate. That you have there listening stations. So yeah, I would search it in the direction of a combination of techniques that together make the puzzle. For if they're mating areas, and yeah.
There was a video, a slide that you showed that sharks had come from the USVI to Saba Bank, which was around Irma time as well. Is it possible they were blown off course, or were they seeking shelter, or does one nothing have to do with the other?
Yeah, the one nurse shark, that was out, but it was quite a bit after. But it could be. We saw also in the sharks in our study that, of course, at St. Martin we lost all the data for that year. But on the Saberbank, for instance, and in Stasia, we saw individuals that. Because we had two, we had Maria and we had Irma with 2 weeks in between. So and we saw that there were more movements in some individuals, and wider scaled movements when Irma and Maria passed, and the rest of the time they were very stationary. So they at least saw part of them really respond to that. But whether if they're really disoriented and move over such large scales due to that I won't say. And the 2 tiger sharks we saw was way after the hurricanes passed. Yeah.
Could we limit it to maybe just one more question? Because I think some people are getting a bit restless. They want to move on to our raffle, and we don't want to stop you from asking questions from Erwin, but we'll ask that you can do that after the talk, and maybe he'll be here for dinner and drinks, but let's have one more quick question.
Sure, yeah.
The individual, I believe it was a reef shark that you tracked around Saba, you saw more sightings during the morning and during the night. Any speculation on where it was during the day?
Yeah, I think we think that because here you also have steep drop-off, so it used also deeper habitats that are beyond the reach of the listening station, because it didn't show up on other stations around Saba. Saba is relatively well covered because it's only a tiny island, so there's not that many white spots in between. Yeah, that's our best guess is that they use a bit more deeper waters adjacent to the reef. For nurse sharks also we saw that because if they really sleep in crevices, and they can also be in even if they're within reach of a listening station, they can be in a sort of acoustic shadow, and but that you can derive from the patterns. You can see that if it's sleeping, you see a very long line, then it's really sleeping within sight of a listening station. But sometimes also you see a gap, and then it's detected one, and then from the sequence of detections you can see if it's in the area covered by the listening station and in an acoustic shadow. Or if it's really outside the area. But for Caribbean reef shark, they swim around. They don't really hide in crevices. So there it's much more likely that they are in deeper water. Yeah. Okay. Yeah. Okay.
All right, now the time that you've really been waiting for. Sorry, Erwin. The raffle draw of the month. So if I can ask my helpers to come up, Mia, Nolan, Jesse, whoever, you can help me pull the tickets so you know it's not biased. So if you want to give it a good shake. And then take it. So first up, our first prize is a Saba embroidery by Emily June, who is one of our Create and Learn artists earlier this year. And the winner is Beth. Phone number 603-289-8801. Congratulations, Beth. Our next prize is a trip to St. Martin, which includes a 2-night stay at Seaview Beach Hotel, a guided hike with Binky, a sunset cruise for 2 with Aquamania Adventures, and a round-trip ticket from Win Air. Want to reach in there? And the winner is Darren Hilong. Congratulations, Darren. Next up is our basket from Saba Greens and Artisanal Treasures from the Saba Reach Foundation. It's a gift bag. So, Mia, you want to do the honors? And the winner is Pam Hollinger. Yeah. Congratulations, Pam. Next up, apparently our slides aren't working, is our Saba package, which includes a 4-night stay at Flamboyant Cottage. Thank you to Larry and Sherry. It includes a guided hike with the Saba Conservation Foundation, a two-tank dive with Sea Saba, and a piece of Saba lace by Peggy Barnes. And the winner is Shea Booba, our sponsor for this evening. Do you want me to pick again?
Please give this to somebody else because I leave.
All right, I will draw again. Want to pick again? Claire. Yeah. All right, we're going to try a third time. Angelita Peterson. All right, our next prize is a hand-painted doorstop by Boomerang. You want it? And the winner is Yuna, Elwin and Corinne's daughter. Next up is our handcrafted knife made by John Magor. And the winner is Tom Walksma, and then it gets cut off. Oh, yay, congratulations, Tom. Next up is a stay in St. Croix. This features 7-night stay at Banana Keat House, as well as 3 days of diving. This is a big one. Pick a good one. The pressure's on. And the winner is Sherry and Larry. Congratulations. Next up is a hand-carved bowl made from West Indian mahogany by Binky Van S. And the winner is Oliver and Nikkei. And our final prize of the evening is a 8-day, 7-night live-aboard dive package on the Caribbean Explorer. All right, who's doing this one? Oh, maybe it skipped. Yeah. Oh, Rule and Roseanne, our experts. And then I guess I'll just say the Stacia package. I'll see if there's a slide. Trip to Stacia. It got skipped in the beginning. Oh, we have 2 more, actually. There's a Sea Saba prize, too. Okay, so the trip to Stacia includes a 3-night stay at Quill Gardens, a guided hike with Stanapa, a 2-tank dive with Scuba Aqua, and a round-trip Makana ferry ticket. And the winner is Tom again. And we also have a discovery dive or an enriched air class with sea Saba. And the winner is Tim Schmitz. All right, thank you everyone for coming and for supporting us. Please stay for dinner, and we'll see you next October.